Yeah, I'm coming from Google. I'll talk about this slide in a minute. We have a cloud. I work in the cloud division. It's great. It has some problems, but it's generally pretty awesome. And I'm going to talk to you about some use cases today. I've been involved in some of this work, but not all of it. I don't know everything about cloud, but I'm happy to help you find answers if you are. I'm just generally here to help you learn some more about what's going on, specifically in life sciences, which I think is a bit unusual. Usually data science people are talking about marketing and time series kind of stuff. And I thought I'd start with this. This is something I'm really excited about right now, this particular slide we've got up. There's a new device coming based on a nanopore, which is this thing here. And there's some DNA you can see flowing through this. It's being driven by an electrical current. And so we're actually measuring the current of the DNA as it's flowing through the pore, and that's this signal here. Based on this, I believe it's a sampling of about 8 kilohertz at 12 bits per sample. We can feed that into an LSTM recurrent neural network and determine the sequence of the DNA at a rate of about 500 base pairs currently. That's limited by the GPU processing. And we can crank it up faster, the voltage, and send that through there faster. This is only one pore, though. These instruments that are doing this typically have, the bigger ones have thousands of pores and are generating gigabytes of DNA sequence per second these days. Which is interesting in a high throughput, like laboratory kind of context. But this device here is pretty interesting. This is, as you can see, it's called a SmidgeIon. That's a little cartridge that has some liquids that get injected into it. And then it plugs into the port on the bottom of the iPhone. And then this thing with an LTE connection, it's only got a single port. That's 100 kilobytes a second, can push data up into the cloud. So we can read data from this thing from a field station. That could be like an agricultural field. It could be somewhere in the Puget Sound. I live in the Seattle area. We've got this problem with norovirus infecting oysters right now. And I didn't get to eat any oysters when I was at the airport. But if we were monitoring the environment, we could catch the outbreak earlier. So we can push this to the cloud, to like a queue, process the data on a auto-scaling Kubernetes cluster, for example, and catch epidemics before they break out, for example. This very similar instrument, earlier generation of it, was used to monitor the Ebola outbreak. And as the Ebola virus was mutating as it was spreading, they were able to watch that in semi-real time and then develop the right vaccine to stop the virus. You can stop poaching type of behavior as well, smuggling, as things are moving through customs. If you can verify that something is what somebody said it is and verify it's not contaminated. Really good application of this too is in the ER. If somebody comes in with an acute infection that's potentially going to kill them, being able to do some sequencing in real time like this can save a few hours, and that can potentially save a life. So this is fun stuff. I'm going to be blogging more about this. Follow me on Twitter, Alan Day. I have a slide about that in a moment with my username if you're interested in learn more about that. Let's just jump into it. Yeah, I'm Alan Day, right? I was already introduced. Let's just go. I've got a lot of slides. I'm at Google Cloud. We do infrastructure. Everything I'm showing you today, maybe not everything, but most of it anyway, is running on Google Cloud infrastructure. And I'm going to give you a bit of background of how to think about precision medicine from a geneticist's point of view, or somebody with a quantitative biology background. It's a bit different than an ethicist's point of view. And I'm not going to talk about ethical issues today. I'm going to talk about technical issues. So please don't snipe me with any pithy, moral kind of questions. Because the technology is going to move forward regardless of what people think in that sphere. And then I'm going to talk about some deep learning applications in genomics and precision agriculture. Oh, hey, OK, emojis. I think I probably don't need to explain too much about what's going on here other than in Android 7.1, we released some professional emojis. This is a girl coder, so we don't just have religious characters anymore. And we all know that people can, we can have some kind of cross-product function that results in some random distribution of children based on the parental types, right? All right, that's genetics 101. And what comes out, maybe some mutations are over in the parents. When you combine them, you create a lethal mutation. Really, though, that baby I was showing you, the product, if we're interested in getting to this product over here and being able to regress back to the inputs. Really, the output is a function of the genotype coming from the parents in the cross function there, and then some environment, which I've here labeled as various baby bottles as food for input. That was the best emoji I could come up with. And we're totally OK with having physicians or other professionals swapped in this situation to guide proper eating behavior and exercise or so on. But we're, in my opinion, it's a bit unusual that over in this crossing function, we're relying on maybe less fact-informed methodologies to make decisions for how to do crosses. If we compare this to chicken love in nature is also a random crossing function. But these are domesticated chickens, and chickens have been domesticated for a while. They also have some food. They don't drink from bottles, but they do eat corn. And usually we're interested in getting out some product from the chicken. It's not going to be a coder, but it would be something more like some delicious rotisserie chicken. And we're totally OK with having artificial selection and influencing of these crossing steps for non-human species. And I'm going to talk about that. We can make dancing chickens. I don't know if this is real or not, but I think we can probably make a dancing chicken. That's pretty reasonable. It's within reach. Usually they're wanting to do something like increase yield, the size of the chicken, and so on. There are these chickens now, I was reading about this, that they make so much meat, so much yield that their legs can no longer support the body. This is what's happening in agriculture. And so over here, this genetic part of it, there's an optimization process, and we call that breeding. Or artificial selection. And then the other optimization step is giving the right kind of environment to this new organism in which you want to cause some kind of outcome. And we would call that environment optimization or adjustment of the phenotype. This is basically the final phenotype we're interested in. And there's some advances that have been happening in, hold on a second, the area of artificial neural networks. Are you guys already familiar with deep learning? Anybody not familiar with this? Because I got a lot of slides. OK, great. Awesome. OK, so yeah, it's a series of tensors. They process stuff. This one I'm showing you here is a convolutional neural network based on mammalian visual system. These functions are, the more of these layers you add, the more of an arbitrary general purpose function you can model. Used to be really slow and not easy to have many layers. But with the recognition that we can use GPUs, we can now do really deep networks. They are also now, a lot of the media and attention is being given to their performance capability on image classification in particular. This is a slide from performance of machines from 2010 to 2016 on a competition called ImageNet, which requires the machines to apply one of 1,000 possible classes to a series of images. And humans have a base error rate of around 5%. Machines are now surpassing humans. This is on general purpose images, but it's also happening on other types of images like medical images. That's just an example of ImageNet. There's all kinds of comedy in here. We don't really have time for it, though. Actually, this one's kind of funny. It looks like a cucumber, but it's not. It's a slug. Yeah. Oh, hey, it's some chickens. The funny thing is though, these machines, they're not only outperforming humans on tasks on which they're trained. So that was, there's a specific network called Inception. It's a specific instance of one of these convolutional neural networks that performs well in the ImageNet competition. You can also do this thing called transfer learning with them. Anybody who doesn't know what transfer learning is? OK, so transfer learning, you have an existing network that you train to recognize some classes. OK? But then let's say you have some other classes you want to extend from 1,000 to 1,002. Or just forget those 1,000 and just give me these 2. The fact that you've put in a bunch of data into the network means that it's able to recognize some general purpose patterns in the earlier levels of the network, and it gets more and more specific as you move downstream. What you can do is something like a micro brain surgery on these things, where you strip off the last one or few layers of the network to get more, to increase its generality, and then bolt on some new layers that are not yet trained, introduce new concepts, and it will recognize them. So all the work you did to train the upstream layers can be reused downstream with only the work required input work goes into training of the downstream layers. So this is a way to move learning from domain A into domain B, which is a very powerful thing. And this is an example of a manuscript. There's a manuscript from 2015 from UCLA. Woo, I went to UCLA. I'm from LA, actually. I lived in Culver City for 13 years. Yeah, I know. Thank you. Thank you. UCLA. Yeah, so they taught it about this fake game called Quidditch from the Harry Potter novels and about what is a T-Rex. And we're able to make that happen. Here's another example of transfer learning. It's not just from image labels to image labels. We can also move the learning between different modalities. So for example, you could take some labels that you had learned about and apply a language model to generate a caption for an image. And that's an example shown down here, another reference from Google 2015. More recently, this is starting to enter the consumer space. You can see there's some kind of interesting artistic filter applied here. This is a product that is now available from Snapchat as a lens that's applying a transfer learning approach to generation of content. So it's the human and the computer are collaborating to make this image. Another example of that is this Van Gogh style transfer, where if we train a machine to recognize Van Gogh style, give it an input image of one of the canals in Amsterdam, we can output a Van Gogh version of the canal. And this is everything I've been showing you so far is being done with this library called TensorFlow. I might have said this earlier. It's now it's the most popular machine learning library and the most popular deep neural network library on GitHub, and it lets you build this kind of stuff. Convolutional neural networks as well as the recurrent neural networks for doing the caption generation or the sequence analysis from the nanopore sequencer I was showing you in the beginning. It also does that kind of stuff, so it's quite general purpose. And it can be used in breeding optimization. So I'm going to talk about this now. I don't know what my next slide is. Marker-assisted breeding. OK, so this is some more biology. If you want to maximize for some particular phenotype of interest, getting back to the chicken, let's say we want it to have more grams of meat per unit time. So it grows really fast on minimal food input. So that's really efficient at making chicken meat. What you would do is you would have some of these organisms. In this case, it's corn I'm showing you here. This is a common organism that's studied. You sample some tissue from it, extract some DNA, generate what's called a marker fingerprint. So you're looking for some molecular markers in the DNA of this individual that you sampled from. And then you do some analysis on this data. And then based on the model that you built of what you had here, if you've got a good predictive model, you can figure out which individuals you might want to cross that will lead to a downstream generation individual of these that is moving in the direction that you're trying to cause that gradient to go in your response variable. And we've been doing this for many thousands of years. We didn't used to do it based on molecular markers. We used to do it based on phenotypes, like, hey, let's make a corn that's bigger and rounder and more yellow and with more starch, for example. This is teosinte. This is the closest wild known relative to corn. It's native to Mexico, and this is a modern ear. It took about 10,000 years to do this, and of course, this is also accelerating, where most of these gains have come from the last few decades, actually. This is an example of traditional breeding. So this is what we used to do. It also increases exponentially as you do this artificial selection. But if you do a marker-assisted selection using genetic markers, the molecular marker, if you can measure the DNA, you can actually get an uplift on that exponent. So we can cause this to exponentially grow faster than it was before. And that's why you see in the literature, this plotted from 1978 to 2008, the increased incidence of this term marker-assisted selection or quantitative trait locus is also increasing exponentially, because that's what's driving the next version of this. This is a slide from an SEC filing from Monsanto. They are #1 in corn breeding and corn optimization. Cartoonifying that marker-assisted breeding slide I was showing you, there's this grow phase, identifying individuals desirable, whether that's via phenotype, like the visible features, or molecular, doesn't matter, and select and recombine. So that's basically the iteration you're trying to do to improve over time. And right, marker-assisted breeding is rapidly increasing the frequency of favorable genes, increasing better phenotypes. So if you want to do this and you want to apply some neural networks, the thing that causes the individuals in a population to not be identical gets back to what I was showing you before of random crossing of the parents, the males and the females, right? There will be some distribution of output progeny from any given cross. But if you don't know what your input variables were, like the actual state of the individuals, you don't really have any way to drive the gradient other than by looking at the observable phenotype. But if we can look at the genetic data and identify places where an individual is variant from an expected value, that's effectively a genetic switch. And that's what this is all about, is how many of these genetic switches can we find so that we can deterministically influence the progeny of a cross. If we want to do that, we would love to use a deep learning machine, but we need lots of examples for biology. And it turns out that because in human, there's about 3 billion base pairs, we have lots of good, lots of observations where we know the true state of a variable independently of any noise that's introduced by our measurement errors or measurement biases. And so genetic data ends up being particularly good at trying to apply deep neural network or deep learning approaches to working in genomic data. Give you a bit of background. This is a genetic data type. What I'm showing you here is a reference genome alignment. So I'll just talk in terms of human for the moment. Humans have an array of about 3 billion positions in length, this being a DNA base pair, so 3 billion base pairs. The genome sequencers, they can't read from end to end as all 3 billion. They can only read little fragments, little shreds of this. And so what I'm visualizing here is these shreds being aligned in ascending order of their start position, aligned to the genome to the end. So each genomic read is a substring, and it's been aligned. We'll do oversampling because the instrument itself has an error rate, and so we want to make multiple observations per column to make sure we got a good read. Because sometimes a given position is not, the true state is actually not identical to the reference. So in here I'm showing you a heterozygote. That's because the individual for whose genome is being sequenced here, one of the parents had a state that was the same as the reference genome, which is a C, and then the other parent passed a T to the progeny. So you can see you've got about 50% Ts and 50% Cs, and you want to be able to call that correctly so that you know what was present in the individual, so you know what's going to get passed to the progeny. Okay, so this is a really clean textbook kind of example. In reality, it doesn't really look like that. It's really more of a mess like this, where you've got all kinds of noise and artifacts and short reads and just a lot of garbage that gets in your way. So you can't really see what's going on. And this is problematic. Of course, you could measure more, but that costs more. Ideally, we'd be able to apply some algorithms to solve this problem. Yay, algorithms. OK, so that's what we did. There was a paper that came out in October of last year from Google. This is a team that I collaborate with, but I was not an author on this manuscript. You can kind of see, I know it's washed out, some of this, deep dream stuff I was showing you earlier, like this slug dog guy and Chewbacca in a kimono. I don't know, like a pawn rabbit thing. In this image here, those are hallucinations coming from some embedded features earlier in the processing of the network before it makes a decision, if we're going to visualize it. What this is a picture of, though, is it's this thing. So we took the data and we turned it into an image so that we could pass it into inception model. The purpose being we want to identify the variants. So giving you some example of what the method looks like, there's a sequencer. This is an Illumina sequencer, not a nanopore. We align the reads, and then from lowest to largest starting position aligned to the genome, and then use an alpha channel to encode whether a given read position as a pixel is identical to the reference or not. So it's more white in this case if it's non-identical. And then the colors are related to the estimated quality of the read and some other things. But we basically encode it down to a PNG. And then feed these, you can see there's a bunch of like low quality reads here. There's a lot of mismatches to the reference, but it's not really clear there's a pattern. That happens sometimes. Feed it into this convolutional neural network as raw pixels from this image. And the output of this thing, if you train it on clearly labeled data, it will tell you whether it thinks something is identical on both strands to the parent, different from the parent on both strands, or a mix of the two. So one is identical and one is different from the reference, with some decent scores. I think this is just the same thing. More examples of that. So I just found this fascinating. If somebody had told me, if these guys had told me they were going to try to do this, I would say there's no way that's going to work. You're taking like some high fidelity numerical data with discrete types of base estimates and a lot of structure in the data with very fine resolution, and you're going to convert it to an image and send it into this neural network that has never seen genetic data before. It's trained on pictures of dogs and cats and cucumber slugs. There's no way it's going to outperform 20 years worth of biostatisticians' time on NSF budgets. But it turns out, like, we entered a competition from the FDA doing a blind sample. I'm not going to talk about the structure of this thing. And whoa. I guess I don't have the slide. This is now like, this is the cutting edge of variant callers for identifying these positions of difference. So we transformed the data into something that is lower fidelity than the input, yet got higher fidelity output, because that's how powerful these deep learning techniques are. I was amazed. The data set that was used to train, it was trained on one human called NA12878. It's a particular individual that is very well studied. So we happen to know there's a sequencing technology that's higher fidelity than this one. It's the gold standard, and we use that to do the sequencing. So we are very confident of where the true positives, true negatives are. Sorry? Yeah, also with oversampling. That's a very well-studied human individual. Other questions, comments? Cool, let's go. Yeah. And these reads and writes and edit capability, this is like CRISPR over here. CRISPR is not the only technology that's going to do what that does, by the way. That's just the first one. First one that's kind of hit general public awareness. There's a whole series of other genome editing technologies. They've just been very expensive and very low throughput. And CRISPR's like very general and very high throughput. And there's going to be more genome editing capability coming out, just like this log linear improvement we're seeing in DNA reads. It's just time shifted a little bit. So there's going to be a whole bunch of gene editing coming soon. And just talking about reads, though, because reads are getting so cheap, it's getting really cheap to generate these marker fingerprints. So if we've got lots of marker fingerprints, we should be able to use that to model the data, right? But remember, it's not just genetic data you need to get a good model. You actually need the phenotype data, too. You need to know how fat that chicken was on the outcome. So what's happening is we're now like, this is no longer the bottleneck. The bottleneck is now over here on building the model. So we need more data and more sophisticated techniques for doing that genotype, environment, phenotype calculation. So now I'm going to talk to you about that. This is a picture of some agronometric data. We all have a GPS. We all know what GPS is now. There's a whole bunch of sensors out there. The National Oceanographic and Aeronautic Association, I forget exactly what it stands for. They collect a bunch of sensor data, like wind sensors and temperature sensors. And this is how we know there's like global warming going on, right? And you can take all these data and get either geographical point values to build like a volumetric a tensor here of those data. And it's a time series as well, right? Incorporating satellite data, drone data in case you can't get high enough resolution data for a particular position. The particular thing that we want to do with all this data is we're trying to get to yield. Just like we were trying to get to chicken meat yield, in this case, it's going to be yield of bushels of corn per hectare, right? So how much density can we get per unit of water and fertilizer and light input? It turns out that it's not uniform for a piece of land. That's what I'm visualizing here. This is a Google Cloud customer. Their name is Descartes Labs. There's another one called Orbital Insights doing a very similar thing. What I'm showing you here is a picture of their corn yield forecast for 2016. So this is visualized as high yield is blue and low yield is in red. It's done by county, and they're using a whole bunch of historical yield data, as well as data in this kind of form that I'm showing you here, including that weather data. They've got something like 10 petabytes of satellite images on Google Cloud. And this is, let's use my hand. This is the interesting part of the plot, though. This red line I'm showing you is the Department of Agriculture's forward-looking estimate in number of days for what is the error in their yield estimate going to be. So they try to tabulate and predict, because they're economists, what's going to happen and how much corn is going to be produced, and that yields to that results in some futures pricing for corn, right? Because we want to know how much is available later, and so we calculate a price on the commodity, and this is a measure of their error. Descartes Labs, by doing this agronometric geospatial approach, is actually able to get consistently lower error in their forecasting model with the backtesting versus Department of Agriculture. And so their primary customer, anybody want to guess who buys from Descartes?
Futures traders.
Woo, yes, it's hedge funds, exactly. Hedge funds buy these data. There's other cool stuff you can do. Like I heard of another example from one of their competitor or company in the same space called Orbital Insights, and they are doing this thing where looking at satellite data of building cities in China, you can actually figure out how different the official reports are versus what's really happening on the ground based on increased area of shadow cast by buildings. So you can figure out how tall the buildings are. So you take the why am I blanking on the total area under the curve, right? Or not the derivative. What's the inverse of the derivative? Thank you. The integral. Yeah, the integral of the space of the shadows to figure out what's really growing and where. And of course, it doesn't match, right? And of course, you can use that to correctly price real estate. Was there a question back here?
Yeah, do you have actual?
Do I have what?
Do you have the actual? Because this is forecast, so can you validate and see how close they actually are?
I don't actually have that data.
But does anybody go back and look at that to see how accurate the forecast is?
Well, it's error. So
It's a plot of error. So less error means more accurate, and of course, as time moves forward, it goes down because it's more constrained, right? Just like I can tell you it's not going to rain in 1 second. Yeah, this is being driven by open source software. TensorFlow is open source. It's freely available. And open access data. So Google was working with NOAA to import those weather data. And we tapped Descartes Labs on the shoulder when the data were available. And they're like, oh, great. OK, we'll now join that table into our data set. So they just query a BigQuery table. Was there a question?
does that include Landsat data?
They're using a bunch of satellite data. I don't know what all they're using. I know a bunch of it, they have to license it. Yeah.
Or even for, like, I thought Landsat is public domain.
Well, you're asking about a public domain set, but that might be not have high enough resolution or have clouds occluding in particular areas. I mean, they want to make a good model. And they've got the people that are willing to buy. So this is pretty interesting because this basically mainlines the weather data, and in particularly interesting is the plants that were present on a particular piece of land is a contributor to the yield that's coming off of that land, right? What is the genetic character at a piece of land determines what the outcome is. And so they found a way to actually correctly price in the genotype of the plant into the financial markets. So that will cause more efficiency and further reduction in risk. It's also, there's a similar trend happening in weather forecasting. Monsanto, who I told you before, is the leader in producing corn yields, corn breeding technology. they acquired a company called Climate Corp a few years ago that started off as a crop insurance company. So they were having better models of weather a few years out, and they were able to offer a premium that was lower to the farmers that allowed them to buy insurance. And then Monsanto came in and acquired that company so that they could be growing the right seeds for a few years out. So they don't have to maintain a bunch of stock that's irrelevant. They just have the stock ready that they know will be useful in a particularly dry year, particularly wet year, hot year, what have you. More signal into what plants to produce. This might even ultimately lead to some kind of climate engineering. I don't know much about that, but if any of you guys know about climate engineering, I would love to talk about that afterward, or email me, please. And this is going to drive development of crop varieties that are particularly optimized for micro conditions. So right now, a lot of corn that's planted on all this acreage I was showing you doesn't have a lot of genetic variety, but in the future it will, because we're going to try to squeeze that risk out, right? Yeah. So basically, we're bringing all the land onto this is from some NPR show that covered this. I just thought it was a cool piece of conceptual art. It's like Internet of Plants. And no, really. OK, so it gets even better than what I've shown you so far. So we know what a GPS is. Tractors, OK, we know what tractors are. They pull things across the ground. Usually there's some kind of thing back there that does stuff to the dirt. Tractor plus a GPS is a printer, a geospatial printer. Here's a map of some terrain. This is the Santa Ana whatever mountains, I guess. I just put this up here as an example of like some interesting spatial features like I was showing you on the earlier slides. But based on this information, you might want to do some pre-treatment of the soil to improve viability and fertility of the soil before you plant some seeds. Then based on seeding, you might want to have some different seed like ink cartridges in here. So as you're moving across the field, you're doing a printing of different types of varieties, so that let's say higher might be more dry and cool, and lower might be more warm and wet, and maybe the effluent fertilizer makes it more salty at the lower altitude. You want to take all that into account as you're doing the planting. And then treatment, by that UAV data, you can actually do some hyperspectral imaging where you're looking at specific areas of visible or near visible spectrum to figure out whether the plant has a disease. And you can frequently see nutrient, if the plants have malnutrition, or if they have some kind of fungal infection or other pest infection, before it becomes visible. So you can then start treating earlier on by using these UAV data. And ultimately, that's all going to result in better yield, or not, in which case you go back in and develop a new strain.
So this one's got to take high resolution. These images
Yeah, frequently the satellite images are, well, it depends on the application. If you're just trying to tell whether something is green or not,
right, that's
yeah, but if you want to be able to pick up like whether there's a fungal infection in a field, sometimes you need very high resolution images.
probably how they get it from the military.
Well, you could have drone fleets accompany the
Well,
tractors.
the drones, yes, that's right. The drones could be this close.
You hear about drone use cases, all these privacy invading applications and things that are sensational to people can worry about.
Yeah.
One of the biggest applications is actually in agriculture, commercial applications today. And it's just, it's going like crazy. Because they have all the cheap genetic data, now they got to get the phenotype data. Yeah, so this terrain, rugged terrain here. Corn originally came from Mesoamerica. This is a map from mapping the diversity of maize. So it'll be most diverse wherever it originally came from, just like there's more human diversity in Africa. Sorry if you don't believe that, but it's true. I know we're data scientists here, so I'm talking to like-minded people. It turns out that corn originally came, they think, from around this Oaxaca region, and that's where the highest density is of genetic variability. That area has a huge amount of variety of corn. I didn't get a number for you of how many, but it's a lot of different types of corn of all kinds of colors and textures and varieties and shapes and everything. The terrain of Oaxaca is also really variable. It has huge altitude changes, huge differences in rainfall, different soil types, you name it, it's there. It's like really, really rugged. And a lot of the reason this variety is preserved here is because there are small pieces of land that are not viable to come in and farm like a big plot of land with the same type of corn. It just won't work. You have to have a special type of corn to fit into that particular special niche of the land to get any kind of yield. So it's still a lot of small subsistence farmers who are living here growing the same corn that their ancestors have been growing for thousands of years. And I expect that what we're going to see, based on all these new signals that are coming into the market to try to drive yields up and risk down, that you're going to see something very similar to what was happening here, albeit on a larger scale, since corn is such a huge crop, of production of lots of different long tail crop varieties that can eke out more yield from a particular piece of land. So we're kind of coming full circle back to where we were with this corn. I want to talk to you about this, cannabis. I think this is really interesting. I have a few things to say. Corn is a really difficult, so I've been an entrepreneur before, and maybe I'll be an entrepreneur again, but now I'm happy at Google, but later, possibly. And I expect there's some people with entrepreneurial urges in the room. Corn is not really a good place to try to build a new product for a small company. It's been worked on for a long time, and it's been had a lot of capital thrown at it for a long time, which gives these large agribusinesses a huge lead. But we have this crop that's that's historically also been around for a very long time. Earliest examples of it go back to about 6,000 BC of it being cultivated in Central Asia. But we discovered the structure of DNA in the 1940s. And this particular crop, economically viable crop, has been prohibited since the 1930s. So not only structure, but all that molecular genetics and marker-assisted breeding and all that stuff I was telling you about on these earlier slides, none of it's been applied here because it all gets started with the federal budgets. And the people who have been in this space are typically, if they're doing illegal things, they're kind of on the fringe of society, maybe not so well educated, kind of doing things in not necessarily the most optimal or way that would be most efficient and rational as we would think about doing things here. So there's this gap where there's a whole bunch of very obvious things to do with this crop to make it sing. And it is a pharmacopoeia. It's a drug powerhouse, this thing. It's not just, there's like 50 different types of cannabinoids and 200 different terpenoid molecules in this thing. Sorry to get into chemistry. Each of them has some therapeutic value. And that's not even counting if you wanted to make some synthetic derivatives off of them. So it's very valuable. I was at the second annual CanMed conference a couple of weeks ago, which is at Harvard Medical School. And there's a whole bunch of physicians there that were coming in and presenting data of what's happening in their local, some were Americans, what they're doing in their local practice and how it's helping patients. They're doing some clinical trials in Israel. And it's just astounding what is being done with this. Like this opioid epidemic happening in the US right now. The cannabinoids are synergistic with opioids. You can drive the therapeutic dose way down and stop all these overdoses that are happening. But we've got this deformation in the system due to federal regulations that hospitals won't allow patients to use it when they're there because it jeopardizes their funding for all the other patients. Where I really felt the writing was on the wall when I was at this conference was these topics where people were talking about neurological disease, particularly ADHD and epilepsy, refractory epilepsy. So these are like epileptic patients who don't respond to anything else. So a lot of times kids have epilepsy. These patients can be treated with cannabis. And so this is basically the federal government getting in between a mom and her kid. That is not going to fly. Yeah. So anyway, I think there's a business opportunity here. Ripe for disruption. I have this whole other set of slides I couldn't show you because I presented it on non-Google time, and today I've got my Google hat on, so I can't get too specific about certain things. But there's no patents in this space yet. In this Open Cannabis project, their whole thing, they recognize that a lot of plant genes have been patented, and they feel that is disrupting, would prevent innovation. there's this interesting thing in patent law where if something's been public domain for a year, it can't be patented. So these guys, they're sequencing basically as much biodiversity as they can and just publishing the data because that starts the clock. And as soon as that clock is running, nobody can patent the gene. Interestingly, yeah, they're like smart. They're pretty smart. They're biologists and lawyers, right? Sticking it to the, I'm not going to say anymore. But, OK, so of course, the left hand doesn't know what the right hand is doing. Patent and Trade Office did issue a trademark last month for this particular popular strain called Gorilla Glue No. 4. The names are fantastic in this area, by the way. So this is basically acknowledgement that cannabis is going to become a commodity federally at some point, and the differentiation is.
Trademark?
They issued a trademark, yeah.
Not a patent.
Trademark for the name.
Trademark for the name, OK.
Yeah. Trademarks really only matter in a space where there's not any way to technically differentiate on technical features, right? It's a marketing thing, which means they recognize it's going to turn into a commodity, like bottled water.
But it's really not that strong.
So it's kind of like, well, why are you doing this when it's, but it's a positive sign. Somebody's thinking, and somebody's not thinking. Production-wise, I think there's going to be something happening where, breeding. OK, breeding is highly fragmented. I think a lot of that's happening because the genetic data have not been available. But like I was just showing you, the Open Cannabis Project is giving everybody the genome sequences of this plant, which is going to do something interesting to breeding. It's probably going to cause more capital to flow into breeding and cause larger breeding centers. But at the same time, because the DNA sequencing and the phenotyping of the plant is getting cheaper, it's going to enable people with three-dimensional scanners and DNA sequencers like that phone one I was showing you to do things at home. So this is just going to be really interesting what plays out here. Distribution, there's a lot of centralization is going to happen here for economy of scale. But I think because of this effect of the breeding not necessarily centralizing, we might end up in a place where we have something like multi-tenant greenhouses, just like you have multi-tenant data centers. I don't know. This is just me speculating. Could be interesting, right? Sometimes there's a barrier and nobody's pollen can get it. Are you a cannabis aficionado? No? But it's interesting, right? Like you've got, so these guys, I didn't include a slide today about Google operating the most efficient data centers, being carbon neutral, et cetera. There's like really obvious applications to greenhouse optimization of these things that we do in our data centers. And that's what got me thinking about, the data centers, the greenhouses for cannabis are going to become multi-tenant. Just my prediction. You heard it here. Maybe.
People I know.
I don't know if you heard it here first, but you heard it here.
People I know in that space are saying already that if you're not into growing at this point,
Yeah.
you probably shouldn't try to get in because the growing space is saturated.
It's too packed. Yet, like everybody is jumping in there. That's not the place to innovate. It doesn't make any sense to me.
But running a bunch of greenhouses for people who are established.
Oh, yeah.
With established customers.
Greenhouse in every house.
There you go. Right. But this gets back to, sorry, I'm testing my slide here. This gets back to this thing here, right? That's what you're pointing out. Yeah, commoditization and branding. That's where it's going. Or everybody can be growing it at home, and it's going to become like a kombucha SCOBY that you keep in your closet.
Can I lease out my rooftop?
I haven't spent that much time thinking about this, but yeah, maybe.
No, I think it
Tweet
makes
me.
complete sense.
Yeah.
I mean, Google doesn't care what I call my website. They host it either way.
But we do want to help send your cannabis business into overdrive.
Yes.
So I've been thinking about this for a while, and it looks more interesting the further I get into it. I recognized that Open Cannabis Project released a bunch of data. I collected it. I collected a bunch of other data for about 1,200 strains. And then after I published, there was like another 300 that were released the same week I published. So this is like the amount of variation available in that project and the public domain in general is just growing exponentially. I pulled in all the public data. I pre-processed it as a public data set. So just like we have that NOAA data set I was showing you earlier, I did the same thing for the cannabis genomic data. I pre-processed it, put it in a database to the point that somebody making an infographic for the New York Times could come in and query it and build something. And it's available. You can go in and use it. BigQuery's got I believe a 1 terabyte free monthly tier. Come in and look at what's in there. Why not? And in fact, we've got people doing this. So I released these data. My colleague, Felipe Hoffa, he's a social media maven, tweeted this out. He's got like quite a reputation on Reddit. So he puts the data on Reddit, a link to my blog post. And then the next day, somebody in /r/datasets grabs it and builds a genome visualization on top of this thing for looking at the variant data. So people are having a good time, and it's distributed, and I think it's all super fun. So yeah, go out there and build what's next. Hope I got you excited about data and cannabis and all kinds of stuff today. Thanks.